The flu is a viral chameleon, changing shape so fast it's hard for vaccines to keep up. Chemical biologist Dennis Wolin says creating a seasonal flu vaccine is a little like viewing a crystal ball to try to decide which flu you're actually going to be fighting off.
That's sort of where the issue is with vaccines, is that they're so specific and good that sometimes they miss the mark if we're infected with another strain of the virus.
And that's why Wolin and his partners at Scripps Research have been trying to come up with a different solution. And a possible solution is a molecule that binds to and disables that part of the virus that allows the flu to enter our cells. Imagine the virus is trying to unlock a door to a human cell and it has a key. Disabling that key is easier than targeting the whole virus because the key and the door lock don't change from one flu strain to the next.
This molecule, what it does is that it's very broadly neutralizing, meaning it can target all the different subtypes of the different flus.
Scripps Research professor Ian Wilson has partnered with Wolin in identifying the therapeutic molecule. You can see it as an orange patch in this picture of a protein spike. The spike is that key the flu uses to open the door to a cell, and the targeted area of it is the same or is conserved in all flu viruses.
The spikes are highly variable, but it's the top part of the molecule here that's highly variable. The bottom part is highly conserved, and that's the part that we're targeting, the part that's conserved among different influenza viruses.
In Wilson's lab, they have crystallized the protein spike and then use X-rays to show its structure. Here you see one of the spikes. The molecule is that green blob in the middle. Another potential advantage is this therapy could be taken as a pill instead of a vaccine shot. Wilson says our history shows a need for better flu protection.
And we like to have something on the shelf for potentially for new pandemics. We're worried at the moment for these new H5 ones, which we've seen that are spreading throughout the world. They started in wild birds, but they've been spreading to mammals.
Testing of the therapeutic molecule has been successful, but so far only in the lab. Still, Dennis Wolin, who now works at Genentech, sees a lot of promise in this idea of targeting the key that lets the virus into our cells.
I think it expands, first of all, way beyond just flu. I think this goes into all different types of viruses. This is providing a new pathway to combating viral infections.
The next step in the research is to see how it works in mice. Thomas Fudge, KPBS News.